The cell cycle is the process in which cell organisms multiply and divide amongst each other. Somatic and sex cells are the two types of cells that divide. In our stop-motion representation of the cell cycle, we will go over each integrative part of both mitosis, meiosis 1, and meiosis 2. Mitosis being for somatic cells, and meiosis 1 and meiosis 2 being for the sex cells. Meiosis 1 and meiosis 2 are both used to increase the genetic variation.
During G1 phase of mitosis, the cellular contents of the cell, excluding the chromosomes, are duplicated. During S phase, each of the 46 chromosomes are duplicated by the cell. During G2 of interphase, a nuclear envelope forms around the nucleus, which at this point contains one or more nucleoli. Two centrosomes reside in the cell as a result of the duplication of what had previously been a single centrosome. Chromosomes are not yet visible individually since they are not condensed. During prophase, chromatin fibers condense into chromosomes that are visible within the cell. The nucleoli then disappear, and each chromosome is made up of two identical sister chromatids that are held together at the centromere. The mitotic spindle starts to form, and asters extend from the centrosomes. The centrosomes will then begin to move away from one another with the help of microtubules. During prometaphase, the nuclear envelope breaks down, and microtubules are permitted to enter the nuclear area. The chromosomes become further condensed, and some microtubules attach to kinetochores. During metaphase, the centrosomes are at completely opposite ends of the cell, and the chromosomes reside at the metaphase plate, along with the centromeres. Kinetochores of the sister chromatids are attached to kinetochore microtubules that will be used to pull apart the sister chromatids. During anaphase, cohesion proteins are cleaved, and each chromatid becomes its very own chromosome when sister chromatids part from one another, forming daughter chromosomes. The daughter chromosomes move to opposite poles of the cell, and their kinetochore microtubules begin to decrease in length. The cell then elongates, while non-kinetochore microtubules also grow longer. Each end of the cell now has its own complete set of chromosomes. During telophase, two daughter nuclei form within the cell, and new nuclear envelopes are created for what will be each new cell. The nuclear envelopes are created using old parts of the parent cell's nuclear envelope. Nucleoli appear in the cell, and chromosomes become less visible, and they become less condensed. Remaining spindle microtubules are broken down within the cell. The plasma divides after the formation of a cleavage furrow, and the cell splits into two. In prophase 1, the DNA coils tightly, and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. In metaphase 1, the tetrads line up on the equator of the cell. The centrosome has replicated, and one has moved to each pole. Microtubules that extend out from each centrosome attach to kinetochores in the center of each side of the tetrads that have lined up on the equator. Anaphase 1 occurs as the microtubules pull the pairs of homologous chromatids toward each pole as the tetrad is divided. The cell begins to lengthen. During telophase 1, the nuclear envelope begins to reform and nucleoli reappear. The cell begins to split, forming a cleavage furrow in the middle. In cytokinesis 1, the cell is finally split, with one copy of each chromosome in each one. Each of the two resulting cells is now 2N, 2C. Prophase 2, metaphase 2, anaphase 2, and telophase 2 repeat the same steps as prophase 1 to telophase 1, with half as much genetic material. Cytokinesis 2 is the final step of meiosis, where each cell splits into two daughter cells, for a total of 4 gametes, each with half the number of chromosomes. Each of the 4 resulting cells is 1N, 1C. There are 4 haploid cells as opposed to 2 diploid cells in meiosis 1. Crossing over occurs again in the 4 haploid cells in order to increase genetic variation.